All right, guys, we're finishing out the last topic 5.6 in this notes packet. And topic 5.6 is really going to address where do some of these conditions that maybe somebody is missing a chromosome or has an extra chromosome or a part of a chromosome, where does that actually happen? In this process of meiosis, can we pinpoint where problems might arise and how that might lead to offspring that have chromosomal abnormalities? And so we're actually going to start with a section of a video. Now, I know it's Amoeba Sisters, and some of you were probably overloaded with Amoeba Sisters last year. We're only going to watch a tiny clip of this to introduce these images that we can take of chromosomes to see if someone might have an extra or a missing or a partial. So let's check this out first, and then we'll come back to the notes.
Would you ever get a chance to see all 46 of your chromosomes clearly? Well, you could in something called a karyotype, an image of all your chromosomes. It's possible to have a karyotype made as part of some types of medical screenings. In a karyotype, the chromosomes are stained and visible. The chromosomes from a karyotype are typically from a cell that is in a mitosis stage, typically metaphase, as chromosomes are condensed and thick. A karyotype during interphase would be a challenge because you don't have that condensed DNA packing. Now, something confusing to clarify. When searching for karyotypes, sometimes it'll be represented kind of like this diagram, and sometimes you see something like this. And while both show 46 chromosomes, the number of chromatids in the two images here seem different. So what's going on? Now, while the mitosis stage is anaphase or telophase would be a time when each chromosome would have a single chromatid, those phases are generally not when you do a typical karyotype. For optimal viewing, a typical karyotype is taken at or right before metaphase. Recall from mitosis that in metaphase, the chromosome would have two joined sister chromatids. It's just that many times the joined sister chromatids of the chromosome can be so close together that to an untrained eye, it might be difficult to see that each chromosome in the karyotype technically has two sister chromatids. When arranging the karyotype, the chromosomes are arranged in homologous pairs. Homologous chromosomes are about the same size and contain the same types of genes, and in each homologous pair, you receive one chromosome from one parent and one chromosome from the other parent. 23 pairs. 22 of the pairs, 44 of the chromosomes, are called autosomes. That means they are not related to your biological sex. They may have genes related to eye color or height, or hair texture. The last two chromosomes are called sex chromosomes because they determine biological sex. Females have two X chromosomes, males have one X and one Y chromosome. Oh, and just a side note, the name X and the name Y have nothing to do with the shape of them. The reason is an interesting topic to look at.
So you could go Google why the X is called the X and the Y is called the Y if you really want to know. But like she said, it really doesn't have anything to do with the.
Would you ever get a chance to see all 46?
There we go. With their shape. Okay, so let's take some of that plus some additional conversation. Let's add it to this last section of our notes. So first, what is a karyotype? A karyotype is really just a snapshot, a picture of chromosomes in a somatic cell. Now, notice I pointed out in a somatic cell. Somatic cells, remember, that's a fancy name for our body cells. These are diploid or 2N, which means, go back to our definition of what makes a diploid cell a diploid. Homologous pairs are still in the same cell. So when I take the snapshot, I can match up or pair up the homologs. And so you see two of each of these. So homologs are represented. So they typically take the images during metaphase, but then there is some computer manipulation. They're going to kind of cut the images out and drag them around and pair them correctly, and then also stage them from big all the way down to small, and then leave this last pair for last on purpose. So this is a normal human karyotype. So karyotypes will show Both autosomes, which the video just said, those are chromosome pairs 1 through 23. They carry genes that code for lots and lots and lots of characteristics, but nothing that really relates back to biological sex determination. The last pair, or the 24th pair, is I'm sorry, these numbers are wrong. That should be 1 through 22, and this should be number 23. The 23rd pair is your sex chromosome pair. So if you have two X's, you are biologically sexed as female. And if you have one X and one Y, you are biologically sexed as male. And this is in humans. So other organisms don't have 23 pairs. A fruit fly, I think, has 4 pairs of chromosomes. So theirs would look a little bit different. But in general, autosomes are chromosomes that carry traits that are not specifically attached to sex determination. The sex chromosomes carry the traits or the genes that will code for, for in men, code for thickened hair and a deeper voice and facial hair in particular, versus female characteristics carried on the X chromosome, breast development and hip development, lighter hair on the bodies. So various traits like that on the X chromosome versus the Y chromosome. We use these snapshots, these pictures, these karyotypes, we use them to see if there's any extra genetic information, any extra chromosomes in a person's body cells, or if maybe we're missing some information. So let me give you a couple of terms. If someone has an extra chromosome, that means instead of being a pair, they actually have 3 of the number 1 chromosome or 3 of the number 9 chromosome. That's a condition called trisomy. See the tri at the prefix meaning 3. So a trisomy condition is if you have 3 as opposed to 2 of any particular autosome or sex chromosome. Monosomy is sort of the opposite. You're missing one. So instead of having 2, you only have 1. There are also things called deletions where a section of a chromosome is missing. And then there are also something called translocations where a piece of one chromosome breaks off and it kind of floats over and attaches to a neighboring chromosome, and it really shouldn't be there. So a translocation is taking a little piece of one and moving it over to a completely different homologous chromosome pair and attaching it in. So before we leave this screen, this is what's in your notes. I would make sure that you understand, maybe circle or annotate the homologs number 1 through 22. Those are autosomes. And then if you have XX, you are female as your sex chromosome, you're a female. If you have XY, you are biologically male. These are the options for sex chromosomes. You have one or the other. Let's look down here. This is a chromosome, or this is a karyotype that shows an abnormality. So take a second and scan it and see if you can spot what the abnormality might be. Notice we have 2 of each. So I'm scanning through here. I'm not missing or I don't have an extra. This doesn't appear so far to be a trisomy situation or a monosomy situation until I get here. So what I have here are not 2 sex chromosomes, which if you were female, you would have 2 X's right here. If you were male, you would have an X and a Y. So really we see a missing sex chromosome. And so this is a condition called Turner syndrome, and we'll talk about that in class when we do our activity Friday. Okay, connecting this back to meiosis, how do these chromosomal abnormalities occur? Well, this is by way of a process called non-disjunction. And so there's your vocab word, non-disjunction. Non-disjunction is a process that occurs that ultimately creates gametes with too many or too few chromosomes. So when those gametes fuse with another gamete, the offspring has too many or too few chromosomes in their starting cell, which then just gets copied and copied and copied and copied and copied. Mitosis, identical copying. So then all of their cells are either missing a chromosome or they have an extra chromosome in it as the embryo grows and as the fetus continues to develop. So where do we find non-disjunction occurring? This is either a failure at metaphase 1, where we're trying to separate the homologous chromosomes equally, or it is a failure at metaphase 2, where we are trying to separate the sister chromatids equally. Either one of those separation events, if the spindle is not formed and attached correctly, the chromosomes will not pull apart equally. And so that is the process of non-disjunction. And you can see over here what we get as end results when we don't separate everything correctly. Like up here, this is an issue with metaphase 1. This pair of homologous chromosomes are doing the right thing, and they're each going to go their separate ways. But this pair of homologous chromosomes, the spindle is pulling both of them back to this side. And so when that cell actually goes through the rest of meiosis, we end up with gametes that have an extra chromosome. This side, this line of cells is going to be missing the chromosome that was given over here. And that's all just because of an accident, a mistake at the spindle that isn't caught in meiosis. And here you can see this is meiosis or metaphase 2. So you see the sister chromatids being pulled apart in metaphase or anaphase 2, really. And in this case, the spindle didn't attach correctly. And so instead of the sisters separating, it gets pulled back over here. We get both sisters in one cell. So the faulty spindle at metaphase 1 or metaphase 2 is really the source of extra or missing chromosomes in this process called non-disjunction. All right, so that wraps up our understanding of a karyotype. We are going to do a project in class on Friday if you're an A day student and Thursday if you're a B day student. We're gonna make a karyotype that will lead to some kind of chromosomal abnormality in the offspring. And so we'll look at seeing if we can diagnose those and then share out a little bit on types of chromosomal abnormalities. So these are not genetic mutations. This is an entire chromosome is missing, or we have an extra one, or it's broken off and floated to a nearby chromosome. Okay, you are done with this section of notes.